Hello and welcome to another episode on the microbial world. Here, we are going to be learning about biosynthesis of biological macromolecules nucleic acids. What is nucleic acid? Nucleic acids are biological macromolecules that store and transmit genetic information. They include deoxyribonucleic acid DNA and ribonucleic acid RNA. Components. Each nucleic acid is made up of nucleotides, which have three parts: a nitrogenous base purines, adenine, guanine; pyrimidines, cytosine, thymine DNA, uracil RNA; a pentose sugar deoxyribose in DNA, ribose in RNA, a phosphate group. Here's a nucleotides linking via phosphodiester bonds forming a polynucleotide chain. Purine synthesis G. The cell builds this large, two-ring base directly onto the sugar. Pyrimidine synthesis T. The cell builds this smaller, single-ring base first, then attaches it to the sugar like prefabricating a shed and then moving it onto a foundation. The DNA switch, RNR. The enzyme ribonucleotide reductase RNR is the single switch that converts RNA building blocks RNDP into DNA building blocks DNDP by removing the two-phosphate O on the sugar, making thymine T. Thymine is uniquely made from uracil U using the enzyme thymidylate synthase. This is a target for anti-cancer drugs because rapidly dividing cells need a lot of thymine. DNA replication is the process by which DNA makes an identical copy of itself before cell division. Key features: semi-conservative. Each daughter DNA molecule has one parental and one new strand. Bidirectional. Occurs in both directions from the origin in most organisms. Template-directed. The parent strand guides synthesis of the complementary strand. Major steps of DNA replication. Step 1. Initiation. Occurs at specific sites called origins of replication. Helicase unwinds the double helix by breaking hydrogen bonds. Single-strand binding proteins SSBs stabilize the unwound strands. Topoisomerase DNA gyrase relieves supercoiling ahead of. Step 2. Elongation. DNA polymerase I adds nucleotides in a 5'3" direction using the template strand. The leading strand is synthesized continuously. The lagging strand is synthesized discontinuously in short fragments called Okazaki fragments. DNA polymerase I removes RNA primers and replaces them with DNA nucleotides. DNA ligase joins Okazaki fragments by forming phosphodiester bonds. Step 3. Termination. Replication ends when replication forks meet or at termination sequences in prokaryotes. In eukaryotes, telomerase extends chromosome ends telomeres to prevent shortening. Studied the image showing DNA replicate. Enzymes and proteins involved in DNA replication. Enzyme, protein, function, DNA helicase. Unwinds DNA double helix, primase, synthesizes RNA primer. DNA polymerase III, main enzyme for DNA synthesis. DNA polymerase I. Removes RNA primer and fills gap. DNA ligase. Joins Okazaki fragments, topoisomerase gyrase. Relieves tension ahead of fork, SSB proteins. Prevent reannealing of strands, RNA biosynthesis transcription. Transcription is the synthesis of RNA from a DNA template. Stages of transcription. 1. Initiation. RNA polymerase binds to the promoter region on DNA. In prokaryotes, the O-sigma factor helps recognition of the promoter. In eukaryotes, transcription factors assist RNA polymerase L binding. 2. Elongation. RNA polymerase moves along the DNA template, synthesizing RNA in the 5'3" direction. 3. Termination. In prokaryotes, transcription ends via row-dependent or row-independent mechanisms. In eukaryotes, termination signals and polyadenylation occur. Post-transcriptional modifications. Eukaryotes. 5' capping protects mRNA and assists in ribosome binding. 3. Polyadenylation. Addition of poly A tail for stability. Splicing. Removal of introns and joining of exons to form mature mRNA. DNA repair and proofreading. Differences between DNA and RNA. Proofreading. DNA polymerase all detects and corrects mismatch bases using its 3'5' exonuclease activity. DNA repair mechanisms. Type, mechanism, example, mismatch repair. Removes mismatch bases after replication. MUTS, muddle system coli, base excision repair. Removes damaged bases, for example deaminated cytosine. DNA glycosylase, nucleotide excision repair. Removes bulky lesions like thymine dimers. UV damage repair. Recombinational repair. Repairs double-strand breaks. RNA-mediated. Differences between DNA and RNA biosynthesis. Feature, DNA synthesis. RNA synthesis, template, both strands replicated. Only one strand transcribed, enzyme, DNA polymerase. RNA polymerase, primer requirement, yes, no, nucleotides. DNPS, NTPS, product, DNA double-stranded. RNA single-stranded, purpose, genetic duplication, gene expression.